PCR refers to polymerase chain reaction. The PCR is a technique aimed to amplify the target DNA fragment to make copies. There are two kinds of PCR, conventional PCR and real-time PCR. About the conventional PCR, there are three main steps involved in PCR technique. The first is denaturation, next is annealing, and the last is extension. In the denaturation, DNA sequence is heated at a high temperature to unwind and separate into two single strands. And in the annealing step, the primer will bind to the complementary region in the DNA template. After the primer already annealed to the template, the DNA polymerase will recognize and bind to extend the new strand by adding new nucleotide base pairing. It is extension step.
Next, I will talk about laboratory equipment and chemical. 1. 10x PCR buffer. 2. 10 millimolar DNTP mix. 3. Oligonucleotide primers. We have forward and reverse primer. 4. DNA template is genomic DNA or cDNA. 5. Tag DNA polymerase. We use magnesium chloride. 6. Nucleate-free water. 7. Thermal cycle or PCR machine. 8. Microcentrifuge. 9. Microcentrifuge tube for 0.2 ml. 10. Vortex mixer. 11. Micropipette P2, P10, P20, and P200. 12. Pipette tip. 13. Rack for microcentrifuge tube. 14. Gauze.
For the preparation of master mix, a large volume of the solution has been added first. In the preparation of the master mix, the total volume has prepared more than one reaction than the required reaction, due to the prevention of micropipetting errors. So, in this experiment, we set up a master mix is 6 reactions, because the required reaction is 5 reactions. For the step preparation of the master mix, firstly, add nucleate-free water 95.25 microliter in a microtube, and add forward and reverse primer is of 15 microtube. 15 microtube of PCR buffers will have add, and 3 microliter of DNTP mix, respectively. Finally, add enzyme tag DNA polymerase 0.75 microliter and mix them. After that, we are record master mix 24 microliter into PCR tube, size 0.2 milliliter, which consists of 4 tubes experiment and 1 tube for negative control. Add the DNA template 1 microliter in the master mix in every experiment tube. The negative control adds nucleate-free water instead of adding the DNA template.
For example, we need to prepare the NTP mix 10 millimolar. To make final concentration is 200 micromolar. In this case, we know C1, C2, and V2. Using the formula, we can find V1. Therefore, we need 0.5 microliter of 10 millimolar DNTP mix solution to complete the final concentration.
Next, I will talk about steps to use a thermal cycler. Step 1. Before operating the thermal cycler, Be sure to read the safety specification and operating requirements. Step 2. Open the cloth and plug the supply power cord into the appropriate electrical outlet. Step 3. Turn on the thermocycler using the power switch on the back panel. Step 4. The thermocycler runs a self-test to verify proper function, and then displays the home screen. Step 5. To open the lid, lift up the lid handle until the lid system opens without assistance. Step 6. Load the samples to the thermocycler. Step 7. To close the lid, push the lid handle down until it stops. Step 8. To create a new protocol, touch New Protocol from the home screen. Step 9. To edit the temperature or time for each step, the sample volume, and the lid temperature, touch the appropriate box and enter a new value in the numeric keypad. Step 10. Touch Run to run the protocol immediately. Step 11. After running the protocol completely, remove the samples from the thermocycler. And Step 12. Turn off, unplug the supply power cord of the thermocycler and cover the cloth. Next, I will talk about maintaining and caring of the thermocycler. The thermocycler should be cleaned on a regular schedule to remove any debris or dirt that might interfere with proper function. Clean the air vents. Remove dust with a soft brush, damp cloth, or vacuum cleaner. Remove any heavy dust that is deep in the vents with a vacuum cleaner. Cleaning a vent allows sufficient airflow for precise thermal control during a run. Clean the outside case and lid of thermocycler. Use a damp cloth or tissue to clean spills off the outside case. If needed, use a mild solution and remove the residue completely. Clean the inner heated lid. Use a soft cloth and water to remove debris and solutions from the inner lid surface. Never use abrasive detergents or rough material that can scratch the surface. Can the reaction block of the thermocycler on a regular schedule to prevent reagents from accumulating and interfering with the ability of the block to change temperature quickly. Always rinse the wells with water several times to remove all traces of ethanol, bleach, or soap. Safety warning labels posted on the instrument and in this manual warn you about sources of injury or harm, which include caution, risk of danger, caution, risk of electrical shock, and caution, hot surface.